Welcome to Study Loop.
Hi everyone, my name is Eric and let's answer problem 30. In this question, we are presented a sequence and a point mutation has occurred. So that means that in the sequence that we are presented, one nucleotide is going to be changed. And the question is going to be asking, how is the polypeptide product changed? Or essentially, the amino acids itself, what is the difference with one nucleotide change? So in order to answer this question, first we have to decipher the original sequence by translating it into an amino acid sequence, and then compare it to the changed sequence. So first, let's start with the original sequence. Now the original sequence reads ATG, and I will be putting them together in triplets for codons to make it easier to break it down into amino acids. So ATG, CGT, TAT, CGG, GAG, and TAG. Now we'll be translating this into amino acids. So ATG is methionine, and then we have CGT, which is arginine, TAT, which is tyrosine, CGG, which is arginine, GAG is glutamic acid, and finally, the TAG is a stop codon. And how I did this was I took a DNA codon chart. You can find it on Google. And all you have to do is follow the chart and match it with the correct amino acid that corresponds to that sequence. As always, ATG is going to be methionine and so forth. Now let's check the changed sequence. Okay, so here is our changed, where we had a point mutation. And that point mutation actually occurs here at the C position. So let's check the new sequence. In this new sequence, we have ATG as normal, CGT, TAT. We have the change from C to G. So here's the new change with the triple G. We have GAG, and we have TAG. Now, as always, the sequence doesn't change, therefore the amino acids should stay the same. So we have a methionine, an arginine, a tyrosine. We'll skip this one here. We'll have a glutamic acid and a stop codon. And so the change, all we have to do is check the sequence for a triple G. And triple G gives us a glycine. And so with the change, we can see that we switched from an arginine to a glycine. So therefore, this is our change in the polypeptide sequence. I hope this breaks it down on how you should approach the problem and to see what the point mutation has caused. Thanks for watching and see you next time.